We experience the world through our eyes, right? But how do we take what we see and turn it into something we can actually measure? Well, today we're going to dive into how scientists are teaching computers to see with unbelievable precision, turning what looks like a simple photo into some seriously powerful data. Okay, take a look at this. These are turfgrass research plots seen from above. Now, if I asked you, what's the exact percentage of living green grass in one of those squares? Could you do it? And would your answer be the same as mine? It sounds simple, but it's a surprisingly tricky visual puzzle. And that's because, for science, a good guess just doesn't cut it. To track things like plant health or how they handle drought, researchers need objective, repeatable data. Our eyes, as amazing as they are just too subjective. We needed a way to take the guesswork out of the equation completely. And that is where computational analysis is a total game changer. We can write a script, basically a set of instructions, that tells a computer exactly what to look for. And the beauty is, it doesn't get tired, it doesn't have an opinion, and it will analyze every single pixel the exact same way every single time. So how in the world do you tell a computer what green even is? You can't just say, look for the green stuff, right? You have to speak its language. And its language is data. The secret sauce here is something called the HSV color space, which is this fantastic tool for zeroing in on specific colors, especially when lighting is tricky. Let's break this down real quick. First, you have hue. That's the pure color itself. Think of the difference between lime green and forest green. Then there's saturation, which is how rich or intense that color is, all the way from a dull gray to super vibrant. And finally, you have value, which is just how bright or dark the color is. It's a way more intuitive system than the standard RGB we're used to. Exactly! That's the perfect way to think about it. It's like having a control panel with 3 dials. You turn one dial to get the exact shade of green you want, another for how rich and punchy it should be, and a third for its brightness. By setting a very specific range on each of these dials, we can tell the program to count only the pixels that fit our perfect definition of a healthy plant. Now, this is where it gets really interesting. Look at how the settings change depending on the conditions. On the left, those are the numbers for photos taken in a lab, under perfect, consistent LED lights. But on the right, that's for photos taken out in a field under natural sunlight. Notice how the saturation and value numbers are totally different? You have to adjust for the real world. That calibration is absolutely crucial. And this brings us right to the heart of the whole process. You don't just pull these numbers out of thin air. You have to meticulously test and refine them. So let's actually walk through what that calibration process looks like, using a real example from this project. All right, here's our original image. You can clearly see there's a mix of healthy green grass and that dry, brownish yellow stuff. Our mission is to create a filter that isolates only the green and ignores everything else. Okay, here's our first try. We set the lower boundary for hue to 21, and the computer says, okay, that's 56% cover. But you can see it, right? It's picking up way too much of that yellowish dyeing material. That's not what we want. So we tighten the filter a bit, we get a little stricter, bumping that lower hue value up to 23. This tells the computer to be pickier, and boom, the cover drops to 46%. You can see a lot of that yellow is gone now. We're definitely on the right track. Let's jump ahead a few steps. Now we've set the hue to 28, and wow, what a difference. The cover percentage has dropped all the way to 21%, and that image on the right is looking so much cleaner. We've managed to filter out almost all of the stuff we don't want. And here it is, our final calibration for this image. With a hue of 30, the program gives us a final answer of 14% cover. This is perfect. It shows how we've zeroed in on only the pixels that represent healthy living grass. This is the precise, objective number we've been looking for. So really the whole point is this. We've gone from a subjective glance to a number we can actually trust. The difference between seeing some green and seeing the right green, it all comes down to a tiny adjustment in just a few lines of code. But look, in science, creating a new method is only half the battle. You have to prove that it actually works. How do we know our custom-made Python script is just as good or maybe even better than the software that's already out there? We have to validate it. It's the million-dollar question, isn't it? We've gone through all this careful calibration, but does it spit out results that we can actually rely on? Let's take a look at the data. So, what you're looking at here is a face-off between our Python method and a standard program called Turf Analyzer, using those images from the controlled lab. Every dot is one photo. If they agreed perfectly, all the dots would fall exactly on that straight line. And as you can see, it's an incredibly tight fit. Now, let's look at the data from the entire field. We're talking hundreds of plots out there in the sunlight. The result? It's even more impressive. I mean, those data points form an almost perfect line. It's a stunning level of agreement between our new script and the established software. And this number right here, this is the proof. It's called the R-squared value. And it basically measures how well the data fits that line. A perfect score is 1.0. Our result? 0.9979. I mean, in the real world, that is astonishingly close to perfect. The method is validated. It works. Okay, so let's zoom out a bit. We've been deep in the weeds with Turfgrass, but the principle behind all of this is what's truly revolutionary. This idea of turning pixels into precise data points is changing countless fields of science. And remember, this wasn't just some fun theoretical exercise. This code was written and this method was validated to analyze the results of this very real experiment, helping researchers figure out how different kinds of grasses cope with stress. But the application of this fundamental idea, it goes so much further than this greenhouse. Seriously, think about it. When a computer analyzes an MRI scan to spot cancer cells, it's doing the same thing, isolating pixels based on their properties. When a satellite tracks deforestation from space, or a drone assesses crop health over hundreds of acres, it's all running on this exact same principle. It's even used for quality control on factory assembly lines. So it leaves us with this really fascinating thought. We've successfully taught a computer to see and quantify the world with a level of precision that can actually surpass our own eyes. The only question left is, what other huge, complex visual problems are we going to point this powerful new tool at next?